Let's talk experimental taphonomy. Now, you remember taphonomy is the process by which fossils are made, right? And so, experimental taphonomy is when you take something that is or was living and you do stuff to it in such a way as to see if you can replicate the conditions of fossilization. Can you cause some of the same molecular and cellular changes? What sorts of things should we expect as an organism dies, dries out, rots, and is slowly turned to stone? Can we model these things in the lab? And the answer is yeah, sure, yeah. So we've got a biofilm formation up there as inspiration for today. All right, so let's have a look. This is a picture showing fossilization, and it's from a lake. So they're taking sediments out of the bottom of the lake, and these sediments are going to correspond to different ages, and there are different layers in there. I think it may freeze over every year, and so you get nothing building up for a while, then a whole bunch of stuff dropping down, at the spring melt, and so you'd be able to read out roughly how many years you're dealing with. You'd also be able to assay the carbon compounds in there and see how old everything is. So they've taken these, they have preserved them, and they are looking at them with some different microscopy, but including Raman microscopy, which we just learned about, didn't we? Yeah. Um, uh, if not, go back and look at the protein taphonomy lecture, and you'll find it. Yeah. Okay. So a lot of photosynthetic organisms will also show up as colored organisms because they are absorbing certain parts of light and reflecting other parts of light. So they're actually absorbing some of that light energy. And so the photopigments are going to appear to have particular colors based on, you put in a bunch of light and they'll reflect a certain wavelength. And so these sediments end up looking just a rainbow of cool colors. And so here is a microbial mat from the bottom of a lake. This is the fresh mat that you can see here. Those are the colors you've got. Here are the little Raman peaks. And some of those they can assign to particular things. Others are a little harder to. Then they go back through some of these different layers. So here is a layer that they've actually carbon-14 dated to 4,530 years ago. So it is partly, maybe totally fossilized, but you can see different little pockets with photopigments in there, and you got these little grains that are making it up. This is basically the sediment that has infiltrated the biofilm. But yeah, you have an annual biofilm that keeps getting sort of flattened, presumably in the spring melt. And you get these little organic layers, like little stripes in there. And in this paper, they actually go through all the different stages between there. But here you can see the process of taphonomy as it happens over just, a few thousand years in a lake. Now, experimental taphonomy is pretty cool because you take living things and then you basically watch them fall apart, but under very specific conditions. You can say, here's what we think would happen to a fish if you left it on a beach. If you've ever been to a beach, have you ever seen a fish that's there? You get them drying out, you get the bone structure preserved pretty well. If other organisms eat into that, they'll typically eat into the belly, so you'll get the sort of a head and the spine part and maybe the tail, because there's not much meat in the tail, preserved the longest. If you were to dissect those, you would see dried out muscle tissues, but they would still retain some of their organization. And these are the changes we should look for in fossils. These are the post-mortem changes of animal bodies, plant bodies, whatever. So, shown here, this is a little photosynthetic microorganism, and here it is when it's alive, looking great. And so they killed these things. They have died, and then they're going to watch them disintegrate, watch them degrade over time. And so after a certain number of days, you're like, okay. So here, while it's alive, you can see individual cells. The cells have organelles in them. They've got a little N for nucleus, a little C for cytoplasm. Here's what happens 3 days after. You get sort of shrinking. The cells are still all together, but they're each individually a little bit shriveled, And you can still see some sort of outlines of organelles, but they look smaller now. They're reduced in size because they've kind of dehydrated, weird as that may be. It's probably an osmotic thing, as the cell wall integrity and cell membrane integrity are lost on death. Yeah, there's basically no reason for the things that were concentrated in the cell to remain so concentrated. And so you get traces of these organelles out. Here you can see some at day 10. Still a little bit at day 17, although this can turn into a lot of breakdown here. But yeah, you can start to see what has to happen. So this is a nicer way to interpret it. The pictures, they could pick out one that looks great, and you wouldn't know if it's representative or not. But here they're showing, what's happening. So, all right, Volvox, which is that nice big green thing with the little tiny green spots inside of it. They're showing what percentage had a nucleus still visible in oxic conditions versus anoxic. And you see anoxic conditions, you get longer preservation of nuclei. You can say, all right, what percentage of them had chloroplasts, where you could still see them, but they were maybe, sort of a thin version. In oxygen-containing environments, those actually survived a little better. Anoxic, they were pretty well preserved, not quite as well. And so on. You can see all these different categories that they were rated in. And so the idea is that you've got maybe 45 days. So this is how long they went. And in 45 days, there was at least a percentage of all of these organisms that still had nuclei and some organelles visible. In some cases, nearly all the organelles, like chloroplasts. Nuclei are fragile, tend to go away, but some plastids, like chloroplasts, tend to survive. So when we look at fossil cells and we see something that looks like a nucleus, there's a possibility that we're actually looking at mitochondria, for example. They would be a different size, but depending on what taphonomy has done to them, it may be hard to tell for sure. So yeah, if you get significant mineral deposition in roughly the first 45 days, that may actually preserve some cellular contents. And so before, people were thinking, wow, does this have to happen? Do you have to get entombed and mineralized within like minutes or within like an hour? But 45 days, that's more manageable. That's a time scale that a lot of different bacteria could work to in coating the sample over with, iron, with pyrite, something like that. So, yeah, very interesting. Here are some examples of cells with organelle fossils in them. And it's hard to say what these organelles are. They could be a number of things. These, I believe, are the Duchanteau fossils. The organelles inside are actually really condensed metal. It's thought that maybe those were some sort of metal storage compartment in some kind of weird metal adapted organism. Really not sure. They don't appear to be nuclei, or at least they don't seem most consistent with nuclei. But you have structures with structures preserved on the surface of cells, different textures preserved on the surface, individual cells still organized in chains the way they were in life, and in some cases with remnants of cellular contents inside, like nuclei. This one from the Miocene looks as though it's got a little chloroplast in there. You can see I think this is the chloroplast. You see the little grana in there. So suborganelle level preservation possible in some cases. But what happens if it's a fish? So here's a branchiostoma. It's a little jawless fish-like thing called a lancelet. And we think it's similar to these guys, which are called conodont animals. And more about those, but they are real common in the Paleozoic and very useful. They have these little teeth that just get everywhere when they die. And these things apparently were worldwide distribution. So they're very useful in dating exactly when your marine sediment comes from. But if you just put this guy in a petri dish, after 2 days, there's very little change in there. And then with artificial seawater in there, so sort of a salty water, but without the bacteria and stuff you would get. And then what you'll see happening is that basically the soft parts on the bottom are the ones that are going to be fugitive. Those are going to go away, but the notochord is going to be preserved because these don't have a vertebral column. They have a notochord, non-mineralized, like cartilaginous little support. And you'll have little blocks of muscles, and those are also going to be condensed up here. So if we're looking for ancient fish fossils, if they took a little longer to fossilize, let's say like 2 weeks, this is what we should expect to find. We probably shouldn't expect to find the rest of the soft parts. If we do find the rest of the soft parts, we know that they probably were fossilized or mineralized much more quickly than 2 weeks, because this is what happens in experimental taphonomy. Cool, huh? Yeah. Here are stromatolites and microbialites. Probably should have shown this back then, but here they are growing. You've got the little cells here, and then they're going to secrete biofilm. That's going to trap different minerals in them. The minerals are eventually going to entomb the little cell inside, which was just trying to get out of the sun, make a little sunscreen. And then it's basically going to end up fossilizing itself. Yeah. Here's a stromatolite when it's alive, a nice green one with lots of those little cyanobacteria on it. What's the smallest thing you can fossilize? Well, who knows, but people have fossilized individual viruses experimentally. So these were put into a sample where there was a lot of silica, and the silica actually deposits on the outside of the viruses. It sticks them all together, glues them together. But after a bit of demineralization, these things were able to be recovered. At least their structure appears to be intact. Nobody has yet found a fossil virus, but you can bet I'd be interested in that. And this suggests that on the right conditions, they are probably preserved. You would need some sort of extraordinary, like a deposition, rather than just like sandstone or something where you're looking for the shape of it amongst the grains of the rock that are there. It's just like an ichthyofossil, basically, in that case. So yeah, could be. Here is another paper, and this is showing some viruses. They're these little hexagon-looking things over here. And what it says is, in this experimental taphonomy study, the protein coats of some of these viruses served as a point where you got nucleation, you got crystallization of some of the minerals from the water. So in this case, it is, I believe, metal ions that are forming on here. So think something like pyrite, for example. And so just like in, if you pour a Coke, you will notice that the bubbles come from particular places, which is where the imperfections are. And it takes imperfections to start to precipitate the gas out of your Coke, or in this case, to precipitate the metal out of the water. And once it starts to precipitate, instead of having a growing air bubble, you're going to have a growing blob of metal. And so there it is. You got viruses which may actually contribute to the fossilization of the organisms around them. And since there are tons of viruses in the air and in the water and in the soil all the time, these little protein-coated bits of life may actually have a more important role in fossilization than we originally realized. Yeah. All right. And it looks like that's the end. So there you go. Experimental taphonomy. Now we're going to try some experimental taphonomy in class, and so that should be fun. Yeah. See what we can do. See what we can replicate. All right. Thanks very much.